Imagine, just for a second, that you are the head of cybersecurity for a massive, highly secure corporate mainframe.
Okay, that sounds like a stressful job.
Right. But to protect this system, you wouldn't just study the one or two computer viruses that happened to breach your firewalls last year.
No, of course not. You'd want to look at everything.
Exactly. If you wanted to be truly proactive, you would download the individual lines of code from literally every computer virus ever made.
Wow. Every single one.
Yeah. You would take each separate snippet of malicious code, run it against your mainframe one by one in an isolated sandbox, and just map out exactly what each line does to your defenses. You'd be building the ultimate comprehensive encyclopedia of hacker tools.
Which, I mean, that would be a monumental shift in strategy. You'd be moving away from simply reacting to an attack after the damage is already done to fundamentally understanding the underlying mechanics of how these attacks are constructed in the first place.
And that is precisely the shift we are unpacking today. But, not with computers. We're talking about human biology. Welcome to the deep dive.
It's great to be here.
So our mission today is to decode how biological viruses act like, microscopic hackers to hijack human cells, and how scientists are building an unprecedented library of their tools to stop them.
Yeah, and the source material for this is just fascinating.
It really is. We are exploring a groundbreaking 2026 paper published in the journal Cell by Fujimura and colleagues. The core asset they built to achieve this is something called the viral ORFeome.
The viral ORFeome, yes.
Okay, let's unpack this. What exactly is an ORFeome?
So to understand the ORFeome, we first have to define the basic unit, right? Which is an ORF, or an open reading frame.
Open reading frame. Got it.
Basically, in genetics, an open reading frame is essentially a specific continuous stretch of DNA or RNA, and it contains the functional instructions to build just one single protein.
Okay, so it's like an isolated blueprint for one piece of the virus.
Exactly, but the researchers in the study, they didn't just look at a few of these. I mean, they synthesized a massive library. We're talking about roughly 13,400 distinct barcoded viral ORFs.
That is, 13,400.
Yeah, it's huge. These represent the individual isolated viral proteins from over 500 diverse viral species.
Over 500 species. So they're looking far beyond just the standard pathogens we hear about on the news, like the flu or COVID.
Oh, far beyond it. They set out to screen the known mammalian viral proteome. So they certainly included sequences from human pathogens, but the critical addition was sequences from animal viruses that are closely related to human viruses.
Right, because of zoonotic spillover.
Exactly. And thanks to modern DNA synthesis technology, they didn't actually need to go out into the jungle and collect the physical dangerous viruses. They just needed the digital genetic code.
Which they could just print out in the lab, essentially.
Right. But DNA synthesis does have physical size limits. They could only seamlessly synthesize fragments up to about 570 amino acids long.
Oh, interesting. So what happens when a virus uses a protein that is much larger than that? Because some of these viral proteins are massive pieces of machinery. Do they just have to, leave those out of the encyclopedia?
Not at all. They developed a very clever mathematical tiling method. So for the larger proteins, they break the genetic instructions down into those 570 amino acid fragments, but they design the fragments to heavily overlap each other.
Oh, okay. How much overlap?
By exactly half. So 285 amino acids. Conceptually, think of it like taking a panoramic photograph by snapping multiple overlapping pictures.
Right, so you don't miss anything in the seams.
Exactly. This ensures that no crucial functional domain of the protein, like no active mechanical part, is accidentally severed in half and lost.
That makes total sense.
Yeah, and then they packaged all these genetic instructions into delivery vehicles and assigned every single fragment a unique DNA barcode. It's basically a microscopic tracking tag so they could mathematically track exactly what each piece was doing later.
Hold on, I want to clarify something about the methodology here because it's important. If we are just synthesizing isolated fragments of DNA and not using the whole living, breathing virus, how do we know these proteins behave the same way they would inside an actual complex infection?
That is a really crucial distinction, and it's basically the heart of the reductionist approach in biology.
Right.
When a virus infects a cell, 100 different things are happening at once, making it incredibly noisy and hard to study single mechanisms. By isolating the proteins, we are asking a very specific question. What is the inherent physical capability of this one piece of machinery?
Right, taking the noise out of the equation.
Exactly. A gear from a watch still has its specific shape and teeth, even if you take it out of the watch. If you put that single gear into a new machine, you can see exactly how it turns the neighboring parts. We are isolating the gears to see what they do to human cellular machinery.
And the reason we need to know what every gear does is all about getting ahead of the next pandemic, right? The threat of zoonotic spillover that you mentioned earlier.
Yes. Zoonotic spillover is when an animal virus mutates just enough to jump into humans. Traditionally, virology has this, well, it's a priority pathogen problem.
Meaning we only study what's already hurting us.
Right. We pour our funding into studying the viruses that are already making us sick, and we study them one by one. But nature harbors a massive silent catalog of animal viruses.
Just waiting out there.
Yeah. If we wait for an obscure bat or bird virus to evolve the perfect molecular key to enter human cells, we are already fighting a defensive war. This systems-level tool allows us to test almost the entire known catalog simultaneously. We can figure out how these proteins manipulate human cells before the virus ever infects a human.
Which brings us to how they actually test this. But realistically, how do you physically test 13,000 separate viral proteins at the same time? You can't have 13,000 different petri dishes running in a lab. The logistics would be impossible.
You don't use separate dishes, you use a pooled genetic screen.
Okay, a pooled screen. How does that work?
So they took those barcoded DNA sequences and packaged them into lentiviruses. Think of these as defanged, hollowed-out viruses that simply act as microscopic delivery trucks.
Okay, I like that. Delivery trucks.
Right. They use these trucks to deliver the entire 13,000-piece viral ORFeome into a massive pool of human cells all at once. And to make sure the effects weren't just a quirk of one specific cell type, they tested this across distinct human tissues.
Like what kind of tissues?
They used colon cells, retinal cells, and melanoma cells.
So they deliver the library into this huge pool of cells, activate the viral proteins using some sort of chemical trigger, and then they just let the cells grow.
Yes. The first major experiment was a simple proliferation screen. They let the cells grow and divide for a couple of weeks. Then they extract the DNA from the entire surviving population and use next-generation sequencing to count those unique barcodes we mentioned earlier.
So the sequencing acts like a roll call.
Exactly like a roll call. If a specific barcode is incredibly abundant, it means the viral protein it codes for made that human cell multiply rapidly. We label those JO proteins.
Because they make the cell go.
Right. And if a barcode is completely missing from the final roll call, it means the viral protein caused the cell to crash, die, or stop dividing. We label those SOP proteins.
And looking at the data, the ratio here is really surprising. They found twice as many SOP proteins as JO proteins.
Yeah, it's a pretty stark difference.
It seems viruses are highly biased toward hitting the brakes on a cell rather than hitting the gas.
What's fascinating here is not just the ratio, but the evolutionary pattern behind it. The SOP proteins, they identified the ones that cause massive structural stress or DNA damage. They are largely universal.
Meaning they work on anything.
Exactly. If a viral protein is inherently toxic, it will crash almost any type of human cell, whether it's a retinal cell or a colon cell. But the JAO proteins, the ones that promote rapid division, they are highly specific to the tissue. A protein that makes a colon cell divide might do absolutely nothing in a retinal cell.
That actually makes intuitive sense when you think about it. I mean, stopping a cell is a blunt instrument.
Yeah.
It's like cutting the main power cord to a building. It doesn't matter what kind of building it is. Cutting the power shuts it down.
Right.
But forcing a cell to grow and divide without killing it, that's like picking a complex lock.
Yeah.
You need a very specific tailored key for a specific door.
That is a wonderful way to visualize it. JAGO proteins, like the HIV NEF protein or the human papillomavirus E7 protein, are precision tools. They have evolved over thousands of years to manipulate the delicate regulatory pathways of the exact tissue they target.
Okay, let's just trace our steps so far to make sure we have this mapped out clearly. The researchers built a massive library of 13,000 viral parts and dropped them into human cells. By reading the barcodes of the surviving cells, they discovered that most viral proteins are designed to bluntly stop cell growth, likely to hoard the cell's energy and resources, while a rare few act as precise keys to make specific cells G and multiply.
That's a perfect summary.
So a question for you, the listener. To ponder, if a virus successfully stops your cell to hoard its energy, it creates a new problem. A stagnant, hijacked cell is going to look highly suspicious to your immune system. How does the virus hide the evidence of its takeover? Keep that in mind, because hiding is exactly what the researchers looked at next.
Right, because once a virus hijacks the cellular machinery, it faces an immediate existential threat from the human immune system.
It needs an invisibility cloak.
Exactly. Otherwise, the body's patrol units will simply execute the infected cell.
And the body's main way of patrolling for these hijacked cells is a complex called MHC class I, right?
Yes. You can think of MHC class I as a microscopic display window, or like a storefront on the surface of every healthy human cell.
Okay, display window. What's it displaying?
Basically, the cell is constantly sweeping up tiny fragments of whatever proteins it happens to be building inside, pushing them up to the surface and displaying them in this MHC class I window. Meanwhile, specialized immune cells called the CD8 T cells act like security guards patrolling the tissue.
So they're walking the beat looking in the windows.
Exactly. They walk by, look into the display window, and if they see normal human protein fragments, they move on. But if they see a fragment of a foreign viral protein, they immediately recognize the cell is compromised and destroy it.
So if you are a viral protein sitting inside that cell, your primary objective is to literally smash that display window, or at least, I don't know, pull down the blinds.
Precisely. To find out which viruses do this, the researchers ran a second massive screen. They put the entire ORFeome library into a fresh batch of cells. But this time, instead of just letting them grow, they used a sophisticated technique involving lasers to physically sort the cells. They used glowing antibodies that stick to the MHC class I windows.
Oh, wow. So they light up the windows.
Right. And as the cells passed through a laser beam one by one, the machine sorted them based on how brightly they glowed. By isolating the exact batch of cells that had 0 display windows on their surface, they could sequence the barcodes inside to see which viral protein was responsible for pulling down the blinds.
That is incredibly clever. And the culprit they isolated in the screen was a protein called MC162R.
Yes, MC162R.
Which comes from the molluscum contagiosum virus. That's the one that causes those benign little skin bumps, right?
That's the one.
But looking at the mechanics of this, I have to wonder, MC162R is a tiny protein. It doesn't actually have the internal molecular tools to degrade a large structure like the MHC display window on its own. How is it pulling this off?
And it acts exactly like a corrupt middle manager.
A corrupt manager. I love that.
Yeah, it doesn't do the heavy lifting or the dirty work itself. It hijacks the host cell's native infrastructure. Specifically, the researchers found that MC162R doesn't just block the window, it destroys the entire display case by dragging it to the lysosome.
And the lysosome is basically the cell's incinerator, right?
Exactly. It's the cellular trash can or incinerator.
But how did they prove the incinerator was involved? That seems hard to track.
Well, they used a specific chemical drug called bifilomycin A1 that completely jams the lysosome, effectively shutting down the incinerator. When they applied this drug, the MHC class I display windows stopped disappearing from the cell surface. They just piled up inside the cell.
Oh, because the trash couldn't be taken out.
Right. That proved MC162R was actively routing them to the trash. And the way it does this is beautifully devious. It hijacks a class of human proteins called E3 ubiquitin ligases.
Okay, let's define that, because ubiquitin is a fascinating mechanism.
It really is. Ubiquitin acts as a molecular trash tag. When a protein in your cell gets old or damaged, your native E3 ligases slap a ubiquitin tag on it, which signals to the cellular garbage trucks that this item needs to be taken to the incinerator.
So the viral protein MC162R physically grabs your human E3 ligases and just forces them to prematurely slap trash tags onto the perfectly healthy MHC class I display windows.
Yes. It weaponizes our own garbage disposal system against us. And the mechanism relies on incredibly precise structural biology.
How precise are we talking?
It relies on just 3 specific amino acids. On the cytoplasmic tail of the MHC class I protein, the part of the window that dangles down inside the cell membrane, there are 3 lysine amino acids. Due to their chemical shape, lysines are the exact physical attachment points for those ubiquitin trash tags.
So to test if this was truly the mechanism, the scientists simply changed the shape of the attachment points.
They did. They genetically mutated those 3 lysines, swapping them out for a different amino acid called alanine, which doesn't have the right shape to hold a trash tag.
And what happened?
When they made that tiny structural change, the entire invisibility cloak collapsed. The corrupt viral manager could no longer attach the trash tags. The display windows remained safely on the cell surface, and the cell was completely visible and vulnerable to the T-cell security guards once again.
That is wild. That shows just how razor thin the margin of error is in evolutionary warfare. 3 tiny amino acids dictate whether a cell lives or dies.
It's molecular combat on the smallest scale.
Okay, let's pull these threads together again. We've seen that viruses must hide from the immune system's security guards by removing the MAC class I display windows. The newly discovered viral protein MC162R accomplishes this by acting as a corrupt manager, hijacking our native E3 ligases to slap ubiquitin trash tags onto 3 specific lysines on the window, dooming it to the cellular incinerator. So a question to chew on for everyone listening. What happens to our medical strategies when a virus uses our own essential garbage disposal system against us? We can't simply turn off the incinerator with a drug because healthy cells need it to survive. If you were designing a therapeutic, how would you target the corrupt manager without hurting the factory?
That is, without a doubt, one of the most profound challenges in modern drug design. You have to find a way to break the specific handshake between the viral protein and the human ligas without breaking the ligas itself.
Exactly. Now, let's look at the third layer of this defense. So MC162R hides the cell from direct hand-to-hand combat with a passing T cell. But what happens if the infected cell realizes it's doomed and decides to sound a systemic alarm to warn all the surrounding healthy tissue?
That brings us to the final major mechanism the paper explores, cutting the alarm wire.
Cutting the alarm. How does the alarm actually work?
When a human cell detects the presence of foreign viral genetic material, its first instinct is to sacrifice itself and release a potent signaling protein called interferon beta.
Okay, interferon beta.
Interferon is released into the surrounding fluid, binds to receptors on neighboring healthy cells, and triggers a massive intracellular panic protocol, effectively locking down the tissue to prevent the virus from spreading.
But the internal wiring required to trigger that lockdown is highly complex, isn't it? It's not just a simple on or off switch.
No, it's an intricate chain reaction. The signal relies on a critical piece of cellular machinery called the ISGF3 complex.
The ISGF3 complex.
Yeah. You can visualize this complex as a specialized molecular vehicle made of 3 parts. It has 2 large structural proteins, STAT1 and STAT2, and a smaller guiding protein called IRF9.
Okay, so 3 parts to the vehicle.
Right. When the interferon alarm bell rings on the outside of the cell, these 3 proteins assemble into the vehicle, travel deep into the cell's nucleus, and physically bind to the DNA to turn on interferon-stimulated genes, which execute the lockdown.
So the researchers ran a third screen to see which viral parts could snip the wires of this alarm system. They bathed the cells in interferon and tracked which ones successfully ignored the panic signal.
And they found 118 viral proteins that effectively block this alarm system.
118.
Yeah, and remarkably, over 60 of them had never been documented doing this before.
That is the power of the ORFeome right there.
Exactly. But they chose to zoom in on one particularly potent wire cutter from the Yaba-like disease virus, a protein simply named YLDV151R.
YLDV151R. And to figure out exactly how it cuts the wire, the researchers utilized AlphaFold 3, right? Which completely shifts how we think about structural biology. I mean, we don't even need a physical electron microscope to see the interaction anymore. We use AI to predict how the 3D shapes physically fold and lock together.
AlphaFold 3 was instrumental here. If we connect this to the bigger picture, the AI modeling revealed that the viral protein physically binds to a very specific pocket on the human IRF9 protein, the guiding part of that 3-part vehicle.
I think the best way to visualize this is like an electrical circuit. So think of the IRF9 protein as the plug and the larger STAT proteins as the socket. To sound the alarm, the plug has to go into the socket to complete the circuit and form the vehicle.
That's a great analogy.
And the AI showed that the viral protein acts exactly like an insulating plastic cap that fits perfectly over the IRF9 plug. By capping the plug, it completely prevents it from ever connecting to the socket. The vehicle never forms, it never travels to the nucleus, and the alarm is permanently silenced.
And just like with the invisibility cloak, the molecular precision here is just staggering. The structural model predicted that this insulating cap relies heavily on just 2 specific amino acids on the viral protein, 2 glutamic acid residues.
So the researchers ran the exact same kind of validation test. They went into the viral genetic code and mutated just those 2 tiny glutamic acids, swapping them for the neutral amino acid alanine.
They changed the shape of the cap, and the result was total failure for the virus.
It just fell off.
Basically, yeah. The mutant viral protein completely lost its ability to bind to the human plug. The cap fell off. The plug smoothly connected to the socket, the vehicle formed, the lockdown genes were activated, and the cell successfully sounded the alarm for help.
Wow. 2 microscopic structural changes completely disarmed millions of years of viral evolution.
It's incredible.
It is genuinely breathtaking to consider the scale of the microscopic war happening inside our tissues every day. All right, let's lock in our final conceptual checkpoint. To stop an infected cell from warning its neighbors, viruses must block the interferon alarm system. The viral protein YLDV14I1R does this by acting as an insulating cap, physically binding to the IRF9 plug so it can't connect to the socket, preventing the alarm signal from ever reaching the DNA.
Spot on.
So think about your own home security system. If a burglar knows exactly which single microscopic wire to snip to disable the entire neighborhood network, the system has a fundamental vulnerability. What is the cellular equivalent of a backup generator? If a virus snips the main wire, how do we medically jumpstart the lockdown genes downstream?
That is the exact question virologists are asking right now. By understanding precisely where the wire is cut, we can develop therapeutics that bypass the plug and socket entirely, basically artificially turning on the lockdown genes even when the virus thinks it has silenced the cell.
We've covered an immense amount of ground today. We've explored how viruses manipulate the blunt force of cell growth, how they act as corrupt managers to throw our immune display windows into the cellular incinerator, and how they physically cap our electrical plugs to silence systemic alarms.
It's a lot of mechanisms.
It is. So when you zoom out and look at this entire library of 13,000 viral proteins, what is the ultimate takeaway? Like, what does this mean for the future of medicine?
It represents a fundamental paradigm shift in how we approach infectious disease. For decades, virology has been largely reactive. A new virus emerges, it makes us sick, and then we scramble to understand how it works and how to build a vaccine.
Right, chasing the threat.
Exactly. By testing over 13,000 viral parts from across the animal kingdom simultaneously, we are mapping the universal underlying grammar of viral invasion. We are learning how viruses speak to and manipulate human biology on a structural level. This ORFeome library is how we get ahead of the next pandemic.
Because we already had the cheat sheet.
Yes. By knowing exactly how these animal proteins manipulate human immunity today, we can design the countermeasures before the virus ever mutates the ability to infect humans tomorrow. We are anticipating the threat rather than chasing it.
We're essentially reading the hacker's source code and patching the vulnerabilities before they ever launch the attack.
Exactly.
But I want to leave you, the listener, with a final provocative thought that builds on everything we've just discussed. If we now possess a physical encyclopedia of exactly how these viral proteins work, if we know precisely the shape of the E3 ligases that MC162R likes to hijack, and exactly the shape of the plug that YLDV151R likes to cap, could we use that against them?
Oh, that's an interesting direction.
Could we synthetically engineer human cells to produce decoy proteins? Imagine flooding the cell with fake, non-functional E3 ligases or dummy IRF9 plugs. These decoys could act as a honey trap, permanently binding the viral proteins, neutralizing them, and turning their own evolutionary weapons into a dead end. If the virus wants to hack the mainframe, maybe it's time we give them a fake server to play in.
That would be the ultimate counterhack.
Thanks for joining us on this deep dive into the viral ORFeome. Keep questioning the unseen mechanics of the world around you, and we'll catch you next time.